Hello, this is Yashveen. In this video, I'll be solving paper 2 variant 2, February-March 2019, 9700 Biology. Figure 1.1 and Figure 1.2 are photomicrographs of sections through the leaves of two different plants. So Figure 1.1 is a photomicrograph of a section through a leaf of Cornish heath named Erica vagans. Figure 1.2 is a photomicrograph of a section through a leaf of Himalayan cedar known as Cedrus deodara. And Figure 1.1 and Figure 1.2 are not shown at the same magnification. A cells labeled A, B, and C in Figure 1.1 and 1.2 each form a different tissue. Name each tissue formed. So tissue A is just below the upper epidermis, so it would be palisade mesophyll tissue. Whereas B in Figure 1.1 is just below the waxy cuticle, so it would be the upper epidermis. And so is B over here, the upper epidermis. Similarly, C in Figure 1.1 and 1.2 would be the spongy mesophyll cell. B. Erica vagans and Cedrus deodara are xerophytic plants. With reference only to xerophytic plants, describe the differences between the leaves of E. vagans and C. deodara visible in Figure 1.1 and 1.2. So if both of them are xerophytic plants, that means both of them would have features of a xerophytic plant. And since this comparison has to be with reference to the xerophytic features, I'll be mentioning the features in the first figure, which is the Erica vagans. So it's got these hair-like structures and a thicker cuticle which is visible, and you can't see much of that in Figure 1.2. And similarly, Figure 1.1 has a leaf which is rolled up, unlike the leaf in Figure 1.2. C. Transpiration is a consequence of gas exchange in leaves. Explain what rate of transpiration is greater during the night than during the day. So during the day, stomata open up to allow diffusion for carbon dioxide into the leaf for photosynthesis, as sunlight is present during the day and is required for photosynthesis. And as a result of stomata opening up, the water vapor from air spaces of leaf diffuse out through stomata. 2. In mammalian red blood cells, carbonic anhydrase has an important role in the transport of carbon dioxide. Carbonic anhydrase is an enzyme, and you're supposed to outline the features of an enzyme. Here I'll write down a very basic answer. So enzyme is a biological catalyst that increases the rate of a metabolic reaction by lowering its activation energy, and it functions by forming an enzyme substrate complex where substrate is complementary to the active site of the enzyme. B. Complete Figure 2.1 to show the reversible reaction involving carbonic anhydrase that takes place within red blood cells. So carbonic anhydrase catalyzes the formation of carbonic acid when carbon dioxide dissolves in water in cytoplasm of red blood cell. C. A ribbon structure of a molecule of carbonic anhydrase is shown in Figure 2.2. The zinc ion is associated with the active site of the enzyme and is essential for the enzyme to function. The molecule of carbonic anhydrase has primary, secondary, and tertiary structure. Explain the extent to which Figure 2.2 shows the primary, secondary, and tertiary structure of the carbonic anhydrase enzyme. So primary structure would be the sequence amino acid, which is not shown in the figure. Secondary structure is formed by the alpha helix and beta pleated sheets of the polypeptide due to hydrogen bonds between our groups of amino acids. So these could be the alpha helix, and tertiary structure would be the folding, three-dimensional folding of already folded polypeptides. And as mentioned in the question, zinc ion is associated with the active site of the enzyme, and active site of the enzyme would have a specific shape, and that shape is formed due to the three-dimensional folding that forms the tertiary structure. And so you could also write about how zinc ion is associated due to the tertiary structure of the enzyme. 3. In 2015, the World Health Organization published the Global Technical Strategy for Malaria 2016 to 2030. The aim of this global strategy, which follows on from 2008 Global Malaria Action Plan, GMAP for short, is to make progress in the control and elimination of malaria. Both the global strategy and GMAP aim to reduce the case incidence, which is the number of new cases each year of malaria, and the mortality rate, which is the number of deaths each year from malaria. Figure 3.1 shows data for the 4 countries in the WHO region. In the WHO Western Pacific region that had the highest proportion of cases of malaria in 2015, for each of these 4 countries, the percentage change in the case incidence and percentage change in the mortality rate over the 5 years period from 2010 to 2015 are shown. Alright, so here's the graph of figure 3.1, and changes on the positive side of the scale show increase in number of cases. So that would be this bar over here, which is shaded gray, and the rest of the cases, as they are on the negative side of the scale, would show a decrease in both case incidence and mortality rate. With reference to figure 3.1, describe the progress made in the control of malaria in 4 countries between 2010 to 2015. Right, starting with the similarities, we can see that the mortality rate, which is represented by the white bars, decreases in all of the 4 regions, and the largest decrease is in Cambodia region, as it has the greatest height, and this is somewhere around negative 94%, so decrease is 94%, since each single unit here represents 4%. So that would cover 2 marks of the answer, and the rest of the 2 marks you'd use for describing the differences. That comes in regards with the case incidence. The case incidence decreases in 3 of the regions and increases in the LAOPDR region, and the greatest decrease in case incidence is in Papua New Guinea, and that would be around 82%, and you can use this piece of data to complete your answer. To all the countries shown in figure 3.1 supplied households at risk of malaria with insecticide treated nets. This is one of the recommendations in the GMAP and the global strategy. Describe and explain the role of ITNS. Insecticides in ITN kill female Anopheles, and so you need to mention how that is beneficial to decrease in number of cases of malaria. Since female Anopheles acts as the vector, killing them would break the transmission cycle of malaria. B. Another recommendation of the global strategy is to carry out rapid diagnostic testing of individuals who may have malaria. This involves testing human blood samples for the presence of proteins specific to Plasmodium. Test sticks can be used, and figure table 3.1 contains information about the 2 RDT test sticks. Test stick 1, where the Plasmodium protein tested for is the PLDH, and this protein is present in all 4 species of Plasmodium, whereas the second test stick tests for the HRP2 protein, which is only present in Falciparum species, which means that if a person is infected with any species other than the Falciparum, the results would be negative, whereas the results would be positive using test stick if the person was infected with any of the 4 Plasmodium species. Some details of the designs of these RDT sticks are shown in figure 3.2. So you've got an area containing mobile monoclonal antibodies labeled with a colored dye, and this is also the region where you would place blood sample. Direction of movement of blood sample is also shown, and here's the test window with immobilized monoclonal antibodies. The immobilized monoclonal antibodies in the test window are not visible. If the blood sample contains a Plasmodium protein that can be detected by the RDT test stick, the mobile monoclonal antibodies bind to one part of the protein, the immobilized monoclonal antibodies bind to the other part of the protein, and a colored line in the test window indicates a positive result for the protein. If antibodies bind to the proteins of Plasmodium species, it means that these antibodies are specific to those proteins. And so these antibodies would have antigen binding sites that are specific to the proteins of the Plasmodium species. One with reference to table 3.1 and 3.2 explain why test stick 1 and test stick 2 will contain different mobile monoclonal antibodies. Because test stick 2, as shown in table 3.1, can detect presence of only proteins of Falciparum species only, it's got antibodies specific to the protein synthesized by this species, which would be the HRP2, and therefore this antibody is different from that in stick 1 because antibodies are specific in shape with different antigen binding sites in their variable regions to bind with and detect presence of different proteins that would have different shapes. 2 blood samples are removed from a person. One sample was added to test stick 1 and the other sample was added to test stick 2. With reference to table 3.1 and figure 3.2, explain what can be diagnosed for this person from a positive test result from test stick 1 and a negative result from test stick 2. So test stick 1 would have tested for all of the 4 species, and if the result is positive, that means any of the 4 species is present in the person's blood. And the negative result for test stick 2 indicates that the protein produced by Falciparum species of Plasmodium is not present, and therefore the infection is caused by a species of Plasmodium other than the Falciparum species. 4. In a double circulatory system, blood passes through the heart twice in one complete circuit of the body. The complete circuit consists of the pulmonary circulation and the systemic circulation. A figure 4.1 is a diagram of the vertical section through the mammalian heart. The differences in the thickness of the cardiac muscles in the walls of the 4 chambers of the heart are shown. So you've got the atria and the ventricles labeled in this figure. Explain with reference to their functions the difference in thickness of the walls of the left ventricle and the right ventricle of the heart. The left ventricle has to supply blood through the systemic route, which is longer and has greater resistance than the pulmonary route. Therefore, left ventricle has to exert more pressure, which is allowed due to the thicker walls of left ventricles. P. The alveolus is the gas exchange surface in mammals. For efficient oxygen uptake, a steep diffusion gradient is maintained between the alveolar air and the blood. Suggest how the steep diffusion gradient for oxygen is maintained at the gas exchange surface. So the gradient or the diffusion is maintained by breathing movements, which brings oxygen into the alveoli that are present next to the pulmonary capillaries, and it is also maintained by the constant blood circulation in pulmonary capillaries. And so blood takes away the oxygen that has just diffused into the capillaries and brings in carbon dioxide from the respiring cells. See, compared to when they were non-smokers, the ability of people who smoke tobacco to deliver oxygen to their bodies tissues is reduced. 2 causes of this reduction include a decrease in the volume of air per breath moving towards the alveoli and a decrease in the ability of red blood cells to carry oxygen. One suggests one reason why smoking tobacco even after only a short time may cause a decrease in the volume of air per breath moving towards the alveoli. Air that reaches the alveoli comes in through the breathing airways, and therefore decrease in its volume means a role of breathing airways. So the reason that is that the lumen diameter decreases as a thick mucus layer and scar tissue might build up when tar in the smoked tobacco damages the cells of breathing airways. To explain why smoking tobacco causes a decrease in the ability of red blood cells to carry oxygen. So tobacco smoke contains carbon monoxide, which binds irreversibly with hemoglobin in red blood cells, and this prevents binding of hemoglobin with oxygen as it decreases the affinity of hemoglobin for oxygen. Human prolactin, or HPRL for short, is a globular protein. It is a single polypeptide composed of 199 amino acids. The protein is transported in the bloodstream and has an effect only on cells that have a receptor cell surface membrane protein known as PRLR. One effect of HPRL is to stimulate cells in the mammary glands to produce breast milk. Cells that have been stimulated by HPRL need more glucose, and therefore the passive uptake of glucose increases. A suggests one reason why the cells in mammary glands that have been stimulated by HPRL need glucose. If they have been stimulated by the globular protein, that means they are supposed to be producing milk, and that would require energy, and energy production needs glucose. B. An experiment was carried out to investigate the movement of glucose and HPRL across whisking tubing membrane. A short section of whisking tubing tied at both ends and containing distilled water was placed into a beaker containing a solution of glucose and HPRL. After 20 minutes, separate samples of the solution in the whisking tubing and the solution in the beaker were each tested for the presence of protein and reducing sugar. Right, let me just draw a figure to visualize this description over here. So this solution has got protein and reducing sugar, whereas none of those are present in the whisking tube, which is this thing over here. So we have a table 5.1. Part 1 asks us to complete the table. Column in table 5.1 headed color after testing. Alright, so first off, the sample is the solution in whisking tube. Method used is the burette solution added to the sample. So we are testing for protein, and the deduction made in the first row is that protein is absent from solution in whisking tube, and that would be indicated by blue color. Whereas in the solution in beaker, protein is present, and so that would be indicated by purple colored solution. Alright, solution in whisking tubing is tested by Benedict's solution when it's added to the sample and the mixture is heated in a water bath. Deduction says it was deducted that reducing sugar is present in the solution in whisking tube, and it is also present in the solution in beaker. That means both of them would have a color somewhere ranging from green, yellow, orange, or red, and so I'd go with red, which would indicate the greatest concentration of reducing sugar. 2. With reference to the deductions made in the table 5.1, explain the movement of HPRL and reducing sugar across whisking tubing membrane. Alright, initially protein was present only in the solution, and at the end it was only present in the solution in the beaker, so it did not enter whisking tubing. However, reducing sugar was only placed in the solution outside of the whisking tube, and after the tests were carried out, it was deduced that it also was present in the whisking tubing, meaning that it moved into the whisking tubing, and that's because it is small enough to cross a whisking tubing, which acts as a partially permeable membrane, whereas protein would be too large to be able to cross whisking tubing element. See outline how glucose crosses the cell surface membranes of the cells of the mammary glands. Because glucose is a hydrophilic substance, it would not be able to cross the barrier of the hydrophobic fatty acid tails, so it would need a hydrophilic or aqueous channel provided by transport proteins, and so it moves by facilitated diffusion. D. The production of milk by the cells of the mammary glands involves the action of several different enzymes. The cell surface membranes of these cells contain the membrane protein PRLR, which would be the receptor. Figure 5.1 shows an outline summary of HPRL involvement in the production of breast milk. Explain why the production of breast milk can be described as an example of a cell signaling process. To answer such questions about cell signaling, you need to outline the stages of the process and all of the components involved. So it starts with HPRL, which is the cell signaling molecule that enters the stream, and PRLR, which is the receptor molecule. So HPRL and PRLR bind together due to being complementary, and this leads to the secretion of second messenger that eventually leads to the response, which in this case is milk production. The secretion of second messenger is not shown over here, so that is not necessary. However, you can mention that in your answer if you wish to. Six plant and animal cells carry out mitosis to form two genetically identical cells from one original cell. A stated the reasons why mitosis is important in both plants and animals. Plants need it for asexual reproduction, and animals and plants both need it to repair damaged tissue by replacing dead cells. Now be careful not to write repairing damaged cells in your answer, because mitosis can only produce new cells and it cannot repair any damages within the cell. However, it can repair damaged cells that are present in the tissue, and therefore it can repair damaged tissues. B. Plant cells require microtubules to form structures that are needed for mitosis. Name one of these structures, and so I'd write down about spindle fibers that are required to separate the chromatids. C. During the mitosis cell cycle, free nucleotides are used for the synthesis of both types of nucleic acid, RNA and DNA. Complete sentences A, B, C, and D to provide information about nucleotides and the synthesis of nucleic acids. Write the correct term in the spaces provided in each sentence. A. Each nucleotide has three components: A dash group, which would be phosphate group, A dash or 5-carbon sugar, which would be a pentose sugar, and a nitrogenous organic base. B. The nitrogenous organic base of a nucleotide is either a protein or a pyrimidine. C. In a DNA nucleotide, the sugar is deoxyribose, and in an RNA nucleotide, the sugar is ribose. D. The synthesis of RNA from a template strand of DNA is known as transcription. D. A virus named Pandoravirus salinus was discovered in 2013 by French scientists. The virus was so large that the scientists initially thought that P. salinus was a bacterium. P. salinus was confirmed to be a virus after further research. 1. List key features of viruses. So viruses have a protein coat, DNA or RNA, and they do not have any characteristic features of a living organisms like growth, sensitivity, reproduction, movement, and etc. 2. The dimensions of viruses are usually stated in nanometers. As P. salinus is so big, it has been described as 1 micrometer long and 0.5 micrometers wide. Convert the width of P. salinus to nanometers. Since 1 micrometer is equal to 1000 nanometers, you'd multiply 0.5 micrometers with 1000 to convert it to 500 nanometers. And this is it. We are done with this paper. Thank you for watching.